Assalamu alaikum. Hello, my dear students. Welcome to another biology class. I'm your biology teacher, Zahidul Hasan, and today we are going to learn some topics from the chapter Plant Physiology. And today we are going to discuss about the second phase of the uh, photosynthesis, and that means the Calvin cycle or C3 cycle, and also the C4 cycle. Okay. So, so C3 cycle and C4 cycle actually, this is the second phase of photosynthesis, and all this cycle belongs to the light independent reactions. Okay, the light independent reactions or dark reactions involve thermochemical reduction of carbon dioxide to form carbohydrates. And this light independent reactions is also known as the dark reaction or Blackman reaction. Okay, dark reaction because in this reaction no light is required. That's why this is the this is also known as dark reaction. And this reaction was first established by Blackman in 1905. That's why this is also known as Blackman reaction. Okay, and this in light independent reaction, it takes place in the stroma of the chloroplast, okay, outside the thylakoid membranes. Okay, so indeed these light independent reactions, mm, it can take place with or without light. For this reason, this is light independent reactions. And these reactions play a key role in producing organic compounds as carbohydrates with the help of ATP and NADH2. Okay, Mm, there are two well recognized paths of these light independent reactions, and the first one is the Calvin cycle or the Calvin Besham pathway or C3 cycle, and another one is the Hatch and Slack pathway or C4 cycle. Okay, let's start with the Calvin cycle or C3 cycle. And this cycle was discovered in chlorella algae by Melvin Calvin and James Basham and Andrew Benson at the University of California in 1947. Okay, by using this, by they use a radio active isotope carbon 14 to discover this cycle, and that's why this cycle is also known as Calvin cycle or Calvin Benson Basham cycle or CBB cycle. Okay, according to the scientist's name. Okay, so the discovery of the cycle, Melvin Kelvin and his colleagues also awarded the Nobel Prize in 1961. Okay, so the first product, the first product of this component of this cycle is 3 carbon 3 phospho sorry, the first product here, this is the first stable product of this Calvin cycle, and this is 3 phosphoglyceric acid. That's why this pathway is also called C3 cycle, due to this uh, 3 carbon compound. This is the first stable compound of this cycle, though the first compound is this keto acid, but this one is the unstable. Okay, so, and this cycle held in C3 plants. Do you know what is C3 plants? C3 plants are those plants which fix and reduce inorganic carbon dioxide into organic compounds using only this pathway, this C3 pathway. So that means the plant that actually mm, use this pathway, this Calvin cycle, is called the C3 plants. So this cycle occurs in the presence of huge carbon dioxide in nature. And the absolute temperature of the reaction of this Calvin cycle ranges from 10 to 25 degrees Celsius, and all reactions of Calvin cycle held within the mesophyll chloroplast. Okay, so the Kelvin cycle, this Kelvin cycle, it has actually 4 phases. Okay, the first phase is called carboxylation. Carboxylation means carboxy, carbon and oxygen. So carboxylation means the addition of carbon dioxide. See, this is the carboxylation step, and in the carboxylation phase, and in this phase, carbon dioxide added with this RUBP. That's why this phase is called carboxylation phase. And then the another phase is phosphorylation phase. You know, the phosphorylation means phosphate addition. addition of phosphate. So when a phosphate is added to a compound in a reaction, then the reaction is called the phosphorylation. So in the first phase of this Calvin cycle, carboxylation occurs and then phosphorylation occurs. Okay, and then the synthesis of carbohydrates. This is the phase of carbohydrate synthesis. Yeah? Mm, After fructose 6 phosphate, it then turns into glucose. Okay, fructose 6 phosphate, it then turns into glucose. That's why this is called the um, synthesis of carbohydrates. And also the fourth phase is the RUBP synthesis, this RUBP. So by this phase the cycle keep continue. Okay, so let's start with the carboxylation phase, that means the first phase. And so this is the first step here, this is the first step of the Calvin cycle, and in this step carbon dioxide from the atmosphere is attached with this RUBP, 5 carbon RUBP, that means ribulose 1,5 bisphosphate, in order to start the photosynthesis. And this reaction is catalyzed by the enzyme which is known as Rubisco enzyme. Okay, natural carbon dioxide enter into a stroma of chloroplast and then combine with this pentose sugar, 5 carbon compound ribulose 1,5 bisphosphate, to form the first chemical compound of photosynthesis. Though the compound is very unstable, it produces temporarily, and this is called the keto acid. This is a 6 carbon compound. The 5 carbon compound RUBP, when it attached with the 1 carbon compound carbon dioxide, and then it produces this unstable 6 carbon keto acid. Okay, and this reaction is catalyzed by this Rubisco enzyme. Here the Rubisco means ribulose 1,5 bisphosphate carboxylase oxygenase enzyme, and Rubisco is the most important enzyme in the world because it is the first enzyme that creates chemical bond between the nature and the life. So this is a very important enzyme. So then this unstable keto acid hydrolyzed with this water very rapidly and produce two molecules of 3-phosphoglyceric acid, 3-PGA. Okay, and this 3-phosphoglyceric acid is treated as the first permanent compound in this Calvin cycle. That's why this cycle is also known as C3 cycle. Okay, and then the second phase of this Calvin cycle, which is the phosphorylation. And in this phase energy reacts with chemicals, okay, to create simple sugar, 3-phosphoglyceraldehyde. So in this phase from 3-phosphoglyceric acid we are going to produce 3-phosphoglyceraldehyde. Okay, so see, 3-phosphoglyceric acid receive one molecule of inorganic phosphate from ATP. As a result, this ATP it turns into ADP, and by receiving a phosphate, inorganic phosphate from this ATP, this 3-phosphoglyceric acid phosphorylated into 1,3-bisphosphoglyceric acid. Okay, 1,3-BPGA. That means 1,3-bisphosphoglyceric acid. And this reaction is regulated by the enzyme phosphoglycerokinase. Here the enzyme kinase, and you know the kinase is the enzyme that catalyzes the reaction of the phosphorylation. Okay, so then this 1,3-bisphosphoglyceric acid, with the help of the enzyme phosphoglycerate dehydrogenase, this 1,3-bisphosphoglyceric acid, it converts into 3-phosphoglyceraldehyde, and here actually NADPH2, actually 1,3-bisphosphoglyceric acid, it reduced to 3-phosphoglyceraldehyde. Okay, and at the dehydrogenation occurs, the NADPH2 removes it, actually it transfers it electrons to 1,3-bisphosphoglyceric acid for the reduction procedure. And here the dehydrogenation, that means hydrogen removal take place. That's why the enzyme is the dehydrogenase. Okay, and this NADPH2 it then become NADP. Okay, and another important thing is one. See here, here are two phosphate. Okay, 1 and 3 bis phosphoglycerate. Bis means 2. That means it has 2 phosphate. So one phosphate is in carbon 1 position and another phosphate is with the carbon 3 position. So see, and but this 3 phosphoglyceraldehyde, it has a single carbon with the carbon 3 position. So one phosphate is removed from 1,3 bisphosphoglyceric acid, and which is catalyzed by this enzyme 3-phosphoglyceraldehyde dehydrogenase. Okay, so one molecule is NADP is produced in this step. Okay, then with the help of isomerase enzyme, this 3-phosphoglyceraldehyde can transform into dihydroxyacetone phosphate. You know, this is the aldehyde and this one is ketone, and both of them are actually isomer because this 3-phosphoglyceraldehyde and dihydroxyacetone phosphate, they have a same chemical formula but just different atomic arrangement. That's why they are isomer. So when this 3-phosphoglyceraldehyde, it converts into dihydroxyacetone phosphate, that means the enzyme should be the isomerase enzyme. So here, mm, triose phosphate isomerase enzyme catalyze this reaction. Okay, so in each round of Calvin cycle, listen very carefully, in each round of Calvin cycle, 6 molecules of carbon dioxide enters into the Calvin cycle and 6 molecules of water, okay, 6 molecules of carbon dioxide and 6 molecules of water enters into the they entered into the Calvin cycle in each round. So in each round of Calvin cycle, 6 molecule of carbon dioxide and 6 molecule of water entered into the cycle. Okay, so as a result, it produced 12 molecules of 3-phosphoglyceraldehyde. Okay, 12 molecules of 3-phosphoglyceraldehyde, and this 12 molecule of 3-phosphoglyceraldehyde, among these 12 molecules, they used in two ways. Okay, only two molecules of this 3-phosphoglyceraldehyde among only two molecules of 3-phosphoglyceraldehyde among 12 molecules are used in the synthesis of carbohydrate. Okay, only two molecules used here in the carbohydrate, and other 10 molecules of 3-phosphoglyceraldehyde are used in RUBP synthesis. Okay, so among these 10 molecules of 3-phosphoglyceraldehyde that used to produce RUPP synthesis, among these 10 molecule, 5 molecule isomerize into dihydroxyacetone phosphate, okay, by triose phosphate isomerase. Okay, so among 10 molecules that produce RUPP, 5 molecules turns into dihydroxyacetone phosphate. Okay, so now the third phase of this Calvin cycle is the synthesis of carbohydrates. That means this portion. Okay, so in this phase, one molecule of 3-phosphoglyceraldehyde undergo condensation with the dihydroxyacetone phosphate. One molecule of 3-phosphoglyceraldehyde condensed with the one molecule of dihydroxyacetone phosphate by the enzyme aldolase, and they produce fructose 1,6 bisphosphate. Okay, and this reaction is catalyzed by the enzyme aldolase. Okay, so this fructose 1,6 bisphosphate then actually here see, so this one then it turns into fructose 6 phosphate. Okay, by losing one molecule of phosphate. Okay, and this reaction is catalyzed by the enzyme phosphatase. Okay, and this fructose 6 phosphate is then converted into glucose. That means glucose, even the starch, after glucose then it polymerized into starch. Okay, so these are the consecutive, actually the this conversion of fructose 6 phosphate to glucose. This is a series of consecutive reactions. Here there are series of consecutive reactions. So this phase of this Calvin cycle is the production of synthesis of carbohydrates, and that means glucose. Okay, so now the last phase of the Calvin cycle is the RUBP synthesis. Here, this RUBP synthesis, ribulose 1,5 bisphosphate synthesis. So in this final step of Calvin cycle, energy and sugar, they interact to form the molecule RUBP. uh, the cycle start again. Okay, see, from here the cycle start again. So 3-phosphoglyceraldehyde and 1,6-bisphosphate, 3-phosphoglyceraldehyde and fructose 1,6-bisphosphate, they combine together to form the ribulose 5-phosphate 5 carbon compound. Okay, through different there are different reactions, there are several reactions take place here, and after this ribulose 5-phosphate production, this ribulose 5-phosphate in the presence of the enzyme ribulose 5-phosphate kinase, this enzyme then get one molecule of phosphate from the ATP, and then ATP after and so this ribulose 5-phosphate receive one molecule of phosphate from this ATP, and ATP then it turns into ADP, and this ribulose 5-phosphate after receiving this phosphate from the ATP, it converts into ribulose 1,5-bisphosphate. Okay, by producing this RUBP, ribulose 1,5-bisphosphate, this start this Calvin cycle again. So this RUBP then re-enter into the Calvin cycle and thus sustain the continuation of this cycle. Okay, so this is the overall scenario, overall phenomenon of Calvin cycle. Okay, now let's talk about the C4 pathway or the Hatch-Slack pathway. In some plants, the first stable product after the fixation of carbon dioxide is 4-carbon dicarboxylic acid. Okay, 4-carbon dicarboxylic acid called oxaloacetic acid or oxaloacetate. Okay, see, this is the first stable compound, which is 4-carbon compound. So that's why this pathway is called the C4 pathway, and the plants actually that use this pathway is called C4 plants. So it was first noticed by Koschak in 1964 in the photosynthesis of sugarcane leaves, but details of the C4 pathway actually were worked out by Hatch and Slack in 1966. That's why this pathway is also known as Hatch and Slack pathway. Okay, so C4 pathway is observed in many plants like sugarcane, maize, and some of the monocot and some of the dicot plants. Okay, so this the first stable product of this pathway after the fixation of carbon dioxide, here the after the fixation of carbon dioxide, the first stable product is the oxaloacetate. So this process held only in C4 plants. C4 plants fix and reduce inorganic carbon dioxide into organic compounds using only this C4 pathway in photosynthesis. The first carbon dioxide receiver of this cycle is this compound phosphoenol pyruvate (PEP). Okay, the absolute temperature of the reactions of Hatch-Slack pathway actually it ranges from 32 to 45 degrees Celsius, and the reactions of Hatch-Slack pathway held within the mesophyll cell of the chloroplast and also the bundle sheath cell of the chloroplast. Okay, so and actually it has two parts. This C4 pathway has two parts. One part, the first part, it take place in the mesophyll cell, and the second part it take place in the bundle sheath cell, and it has two carbon dioxide fixation steps. One carbon dioxide fixed here in this mesophyll cell phase, and another carbon dioxide fixed here in the bundle sheath cell. Okay, so now let's talk about the first part in mesophyll cells. The first carbon dioxide fixation occurs here. Atmospheric carbon dioxide then combines with 3-carbon phosphoenol pyruvic acid. It combines with the 3-carbon phosphoenol pyruvic acid to form the first stable product oxaloacetate. And this reaction is catalyzed by the enzyme phosphoenol pyruvate carboxylase. Okay, and then this 4 carbon oxaloacetate, then this 4 carbon oxaloacetate is dehydrogenated by NADPH to malate. Okay, after the dehydrogenation, this oxaloacetate turns into malate, and this reaction is catalyzed by the enzyme malate dehydrogenase. See here the NADPH, it gives its proton to oxaloacetate. Okay, as a result, this oxaloacetate turns into malate. Okay, so this malate molecule, and in this reaction, see here NADP is produced. Okay, so then the second part of the C4 pathway start here. It started here in the chloroplast of the bundle sheath cell. 4 carbon malic acid or malate, it then undergoes decarboxylation. Okay, it then decarboxylation in presence of the decarboxylase enzyme, and NADP to form carbon dioxide. Here the NADP turns into NADPH. So that means NADP reduced to NADPH here in this phase. So the second phase, the carbon dioxide in the in this phase of the C4 pathway, the malate it turns into pyruvate by the enzyme malic enzyme. Okay, and here in this stage of the Calvin cycle, the carbon dioxide released in decarboxylation, had see one carbon dioxide is released here, and it combines with the this carbon dioxide which is released here. It combines with the ribulose 1,5 bisphosphate of the Calvin cycle, and actually it enters into the Calvin cycle. Okay, so the pyruvic acid produced, then the pyruvic acid which is produced from the malate in this phase is then transported back to the mesophyll cell, and here it is converted into 3 carbon phosphoenol pyruvic acid. That means, see here the pyruvate, it transported back here in the mesophyll cell, and then it again turns into the phosphoenol pyruvic acid in the presence of the enzyme pyruvate phosphate kinase. Okay, so it again made available for the C4 pathway. Thus in the C4 plants, the initial few steps are different than the Calvin cycle. So but later on the reaction, see here the Calvin cycle is also attached with this C4 pathway. So that means C4 plants have both the C4 and C3 pathways. Moreover, in C4 plants, carbon dioxide fixation occurs twice. Okay, first in mesophyll cell with the phosphoenol pyruvate, and then again in the bundle sheath cells with RUBP. That means in the Calvin cycle. So for this reason, the C4 pathway is also called the decarboxylation pathway. Okay. So I think this is all about C3 and C4 pathway. Okay, now you have to, here is a sample CQ question, so you have to answer these questions from this stem. This is a stem, and see in this stem, this is the overview of photosynthesis process, the photophosphorylation, that means the light dependent reactions, and here the light independent reaction. Okay, then it produces sugar, carbohydrates. So you have to answer these 4 questions, and their mark is 1, 2, 3, and 4 respectively. So you have to define what is light independent reaction, and then write down you have to write the differences between C3 and C4 plants, and you have to describe the pathway in which first stable product is oxaloacetic acid. Which one? The oxaloacetic acid is the first stable product of the C4 pathway. That means you have to describe C4 pathway here in the question C, and then the question D is you have to explain in this question. You have to explain how carbon dioxide converted into sugar in the process of photosynthesis. See how carbon dioxide converted into sugar. So the pathway is the Calvin cycle, okay, or C3 cycle. So I think that's all for today. Thank you all.